Okay, good. So the acid-base balance, as I was saying in chapter 27, is the little talk I want to give on that is going to help you in both lecture and lab. All right. If at any time that you can't hear me, I've been holding a lot of these meetings and my internet is kind of sketchy every once in a while. I guess if you have a mic, I know Lena does. Kimberly has a mic, but she's muted. But if you can't hear me for some reason, Lena, maybe you could say something, or you could type it in the box.
Okay.
Because several meetings, I just kept talking, and I didn't know people couldn't hear me. So try and chime in if it goes bad. The other reason why I wanted to do the Zoom instead of the conference that we've been doing in Canvas is because with the final exam that's going to be coming up in both lecture and lab, students are going to be required to use that Responda software, the lockdown browser, to take the test. Everybody who has Respondus working on their computer is going to be fine. They're just going to take the test when I open it, and it's going to be due by a certain date, and it's going to be okay. But there's a few people that cannot get Respondus to work on their computer, and in which case I'm going to have to schedule a block of time where we have a Zoom meeting, and the students without Respondus are going to have to log into the meeting in order for me to proctor their exam live. So that's why I wanted to practice with this Zoom as well. All right. So with that said, does anybody have any questions? And I'm going to post information in Canvas about all this stuff as well. Do you guys have any questions specifically that you may want to ask before I start?
Somebody,
when is the final? Well, all right. So I invited two different sections in here. The section, depending on what section you're in, the allotted time for the Zoom meeting will be during the regular exam scheduled time, which I have to go look at. But everybody that has Respondus, I'm going to open the test on the 5th, May 5th, and you have until May 8th, which is a Friday, that Friday of that week, 2 weeks away. From May 5th to May 8th, everybody with Respondus will be able to log in at their convenience during those days to take the test. Everybody who does not have Respondus or can't get it to work is going to be invited to a specific time to take their test on the computer. And I have to look that time up, like the 9:30 to 10:45 class, Tuesdays and Thursdays. It's whatever time is in the exam schedule for that class is the 2-hour block of time I'm going to make a meeting. So yeah, I'm going to post all of that, Kimberly, and announcements in Canvas as well, probably sometime later today when I get done with all of my classes. So everybody's going to have the information that they need. It's going to be fine. All right. So with that said, then let's get started in the urinary system. So I can get into the physiology of some of the stuff that I wanted to cover today. All right. First of all, we're not going to be identifying any pictures on the lecture test or the final, but I'm going to use the pictures obviously to teach from. So the gross anatomy of the urinary system is real simple. You have two kidneys. Everybody knows that. There are two ureters that lead from the kidney down to the bladder, carries urine from the kidney to the bladder. The bladder is a temporary storage site for urine. And then when we go to the bathroom, urine exits the body in a urethra. Pretty simple. The physiology that we have to talk about deals with what's going on really inside the kidney, specifically with this little yellow tube looking structure here. So this little yellow tubular structure that you see here is called a nephron. There's 1 million of these in each kidney. They're microscopic, and this is the functional unit of the kidney. These structures allow the kidney to regulate all aspects of the blood. So a couple of slides up, you can look at what the kidney does. It regulates ion balance, electrolyte balance in the blood. It's involved in regulating our blood pH, regulates water volume in the blood. It, excretes waste products out of our body. So the structure that's doing all that in the kidney is this nephron. All right. So there's 1 million of these in each kidney. I want you guys to learn the blood flow through the kidney. That is in the video lecture. I don't know if you viewed it yet, but this is fairly straightforward. We're not in lab. We have to be able to identify these vessels, but not in the lecture. But I still want us to know the order of the vessels in which blood is coming into the kidney and then back out of the kidney. And that's what this little flow chart is for. So if you guys have any problems with this, because you can't really see a whole lot right here, you can actually see them a little better if you have lab on those learning materials and the Quizlets that I made in there on the kidney. But if you have a problem with this, just let me know and I'll go and explain it. Well, we could do a one-on-one even. All right, so as far as the nephron is concerned, it's really composed of 2 parts. The circular structure here, which is called the renal corpuscle, and a tube. This tubular structure is just called the renal tube. The renal tube has several parts to it that we have to really identify in lab and also know what's happening in the physiology. So the first part of this tube is called the proximal convoluted tubule, and it then goes into a loop called the loop of Henle or the nephron loop. There's a descending limb, there's an ascending limb, and then you get into what's called the distal tube or the distal convoluted tubule. So we have to look at the physiology, what's going on at the renal corpuscle, along with what's going on with the fluid in this tube. And notice there's arrows on the tube. That means the fluid that is going to be formed from filtering the blood only flows in this one direction. And that's going to be good for you as well in lab, because in order to identify the parts of the tube, if you always start identifying the tube from where it connects to the renal corpuscle, you'll never get it wrong, because the tube that connects to it is called proximal. It's called convoluted because it twists all up. And then there's a loop. You always know the downside and the upside. If you're starting from the proximal tube, the fluid can only move in this direction, descending and ascending nephron loop. And then we get to the distal tube. So what I want to do is talk about what goes on at this renal corpuscle and what this glomerulus is. This is actually the filter. It's a capillary bed inside the renal corpuscle. So this is where all of our blood is filtered in the kidney. Here's an enlarged picture of it, in fact. So you can see here some detail. Number one, this little artery called an arteriole that brings blood into these capillary loops. This is called the afferent arteriole. So the afferent arteriole is the most important arteriole in the kidney. And I'm going to tell you why when we get there in a second, but and I'll probably have to come back to this picture, but the afferent arteriole carries blood in. All the blood inside the capillary loops is under a high pressure, and it forces fluid and solutes out into what's called the intercapsular space. So there's a capsule that surrounds the glomerulus. So the fluid that comes out of being filtered out through the capillary bed and the wall of the filter is called filtrate. The chemical composition of filtrate is pretty much identical to blood, except filtrate never is supposed to have red blood cells, white blood cells, and platelets in it. And it's never supposed to have a lot of protein, large proteins, and even medium-sized proteins. All the blood cells and platelets and the protein gets held back. They're not supposed to be filtered out. But a lot of your water comes out, your electrolyte, all those ions, vitamins, other nutrients like sugar, glucose, and then waste products all come out. So we're filtering out bad things, but we're also filtering out good things that become part of this fluid we call filtrate. And then that filtrate has to move through the renal tubule. So the first part of the tube, remember, is called the proximal convoluted tubule. And then the filtrate is going to move along the tube. And as the filtrate moves along the tube, the epithelial cells that line the tube have the job of reabsorbing anything that's good that we filtered out and putting it back into the blood. And we have a second chance of getting rid of substances that bypass the filter. So the blood comes in. Yeah, we're filtering, but blood's also leaving via the efferent arteriole. So some things are leaving that we really want to get rid of. So we have a second chance of getting rid of that. That's part of the physiology I'm about to show you. So here is the filter. If we take a section through the glomerular capillary, there's different layers to it. There's 3 layers to the filter. Each layer has the job of blocking some substance from leaving the blood. So I know this is weird. It looks like Swiss cheese, but this represents a special capillary bed that forms the glomerular loops. This is called a fenestrated capillary. So the fenestrated capillary allows pretty much everything to be filtered out through these little holes except our blood cells and platelets.
All right.
Yeah, go ahead. I didn't see who was talking. If you have a question, you can interrupt me if you want. So the first part of the filter holds back all of our formed elements that we learned about in chapter 19. The second part of the filter is the basement membrane that surrounds the capillary. It has the job of blocking large proteins from leaving, like the clotting protein we talked about in chapter 19, fibrinogen, albumin, and a whole bunch of other proteins. They don't pass this part of the filter. So they're held in the blood. So we're not filtering it out. The last part of the filter is this little, it's not really maroon or reddish color in there, but that's how the artist drew it. This is called a slit membrane. The slit membrane is formed between these little cytoplasmic extensions that come off of this weird cell. So if you notice on these capillary loops, there are these weird cells that adhere to the surface of the capillary itself. And from one of these, called a podocyte, to another one, they have interlocking finger-like projections. Those are called pedicles. In between the pedicles is where the slit membrane's at, and the slit membrane blocks the medium-sized proteins from leaking out into the filtrate. So we can have very small proteins come out, your vitamins, amino acids in the blood, your sugar comes out, and things like that. So that's how we filter the blood, by basically preventing things from leaving the blood based on size. How big the substances are is what holds them back. All right, so here's a couple of nephrons. We have a cortical nephron. This is the majority of all the nephrons in the kidney. There's about 80 to 85% of all nephrons are what we call cortical. The cortical nephron has its renal corpuscle higher up in what's called the cortex of the kidney. That's more towards the surface of it. And it has a fairly short loop. So its loop of Henle is rather short. On the other hand, though, about 15% of all the nephrons in the kidney are these types. And this is called a juxtamedullary nephron. So they have their renal corpuscle closer to the border between what is called the cortex of the kidney, the outer part, and what's called the medulla, or the inner part. This is called the corticomedullary junction, or line. And so their renal corpuscles are closer to this line than cortical nephrons. But the main difference is that they have a longer loop of Henle. So they have a greater capacity to concentrate urine. But for the most part, is the physiology that we're going to cover, I'm going to state that the physiology is the same in both of these nephrons. All right. So let's look at how the kidney, or really a nephron, makes urine. Because basically, urine is the production product of the kidney. Everybody knows that. Your kidneys produce urine. But how do the nephrons in the kidney make the urine? And so there are 3 separate physiological processes that have to take place before urine can be produced, and thus we can get rid of unwanted substances from the blood. Maybe if we have too much water in the blood, we can get rid of excess water. excess electrolytes, whatever and waste products. In order to get rid of those substances, we have to go through 3 processes. Number 1, we have to filter the blood at the glomerulus. So here, this is a very simple diagram representing the afferent arteriole, the glomerular capillary loops, which they just show 1, and then the efferent arteriole that carries the blood out. Now, the efferent arteriole carries the blood out of the glomerulus, and it goes into one of 2 capillary networks. So that's what's really shown here. This is called the peritubular capillary network if it's up in the cortex. So up here from the efferent arteriole, you can see part of it goes down in the medulla. This is a capillary bed that comes from the efferent arteriole. In the medulla, this is called the vasa recta. But up in the cortex, where it's encircling all the tube in the cortex, it's called the peritubular capillary network. Now, the importance of this is that the capillaries surround the renal tubule all over the place. So you can see it down here around the loop, up here around the proximal tube and the distal tube, so forth and so on. And they don't show it around the collecting duct and all, but it does go around there. So, oh, where was that picture at? Oh, here it is. So that's what this right here represents, the capillary network that would encircle the tube either in the cortex or the medulla. So here's what's important about that. Number 1, we filter the blood at the glomerulus. That's called glomerular filtration. So obviously we have to filter the blood in order to take stuff out of it. But since we, oh, did I go out? Can you all hear me?
Yeah.
Somebody just typed in, did you go out? Oh, now I can hear you. That's Kimberly. Okay, good. All right, so obviously we filter the blood, but we're filtering good stuff and bad stuff. Remember, we're just filtering based on size. So anything that can fit through those 3 layers of the filter is going to be forced out. So all of the good stuff that we filter out, we have to take from the inside of this tube and put it back in the blood where it came from, someplace distal from filtration. So that's what this peritubular network in vasa recta is all for. It encircles the tube so that the cells that line the tube can reabsorb all of our good nutrients back into the blood that we need. On the other hand, the third that's called tubular reabsorption. So we have to reabsorb all of the good stuff that we filtered out. On the other hand, some things will bypass the filter because the blood's going to be coming in and leaving. So if there's anything in the blood that we still need to get rid of, all the way down the length of the tube, we can take stuff directly from the blood and secrete it back into the tube, and that's called tubular secretion. So these 3 processes has to happen in order for urine to be produced. And when urine is produced, that means the kidney regulated all aspects of the blood. We got rid of waste products. We got rid of excess material. We either had to conserve water if we were dehydrated, or we had to lose water if we're overhydrated, things like that. So by doing these 3 steps, filtering, reabsorbing, and secreting, we actually produce urine and thus maintain a good, healthy concentration of everything in the blood, including water and electrolytes. So we need to look at how the filtration is regulated. So here is the renal corpuscle again. We have to talk about how we're going to regulate the rate by which we are filtering. And there's going to be some questions on how this regulation occurs. So in order for the blood to be filtered at the glomerulus, the pressure inside of the glomerulus has to be high. It has to, in fact, this is a very specialized capillary bed, more so than other beds in the body, because the blood pressure in this capillary bed is higher than any other capillary bed in the body, number 1. Number 2, it's the only capillary bed in the body that has an arterial feed and an arterial drain. So most capillary beds have an arterial feed, but a venual drain. This one is very special, and it is specialized for filtration only. So there are 2 basic things that have to happen in order for us to be able to filter blood out of the renal corpuscle at a constant rate. We have to maintain a constant blood pressure in here. That's number one. And we can alter the surface area that's available through the capillary wall for filtration. So I'm going to describe the second one after. We're going to do the blood pressure first. So how do we control blood pressure in the glomerulus? Well, it's fairly simple. We change the volume of blood that enters the glomerulus. Excuse me. So the way that we do this is this. The afferent arteriole is always larger in diameter normally than the efferent arteriole. You can almost see it on this picture. So since we have a larger input of volume of blood than an exiting point of blood, pressure builds up in the middle. So basically, you're letting a larger volume of blood come in than leave over time, so the pressure builds up in the middle. But since pressure is what is exactly driving the rate at which we're filtering the blood, excuse me, then if we alter the blood pressure in here, we're going to alter the filtration rate. Now, the reason why the rate at which we filter the blood is important is this. So let me go back up to this picture of the nephron. If we are, for some reason, filtering way too quickly, that means it pushes the filtrate through this tube too fast. And if we're filtering too quickly, the filtrate moves too fast through the tube, and you don't have time to reclaim all of your good stuff that you need because you're bypassing all the transporters and you're dumping it out in urine. On the other hand, if we filter too slowly, if whatever's changed, the pressure goes down in here and we filter too slowly, the filtrate moves through the tube too slowly and you have too much time to reclaim stuff. And so you're reclaiming too much salt and too much water and other things, and you're not excreting the stuff we need to excrete out in urine because we're reabsorbing it. All right, so I hope that makes sense so far about why we have to control how fast we filter. So again, talking about the blood pressure in here, blood pressure is exactly what drives filtration. So the pressure inside the glomerulus is called the glomerular blood hydrostatic pressure. And we can alter the pressure in here by changing the diameter of the afferent arteriole. If we vasodilate this afferent arteriole and make it bigger, more blood volume will enter the glomerulus and make the pressure go up. And if the pressure goes up, filtration rate then increases. And then the filtrate starts moving through the tube more quickly. So if for some reason the filtration rate was too low, we would want to vasodilate this arteriole and thus cause more blood to come in, increasing pressure and then increasing filtration rate back to normal. On the other hand, if the filtration rate is too high and we're filtering too quickly, we want to vasoconstrict this afferent arteriole and allow less volume of blood to come in, which decreases the pressure in here, and your filtration rate goes back down to its normal level. So that's what I'm about to try and explain to you. All right. So let's look at, and that's what this slide talks about, if it's too high or too low. So let's look at glomerular filtration rate. And there's some animations in here you should go look at if you haven't done that yet. On average, we filter about 125 milliliters of blood or produce 125 milliliters of filtrate every minute. Females are a little lower, but nonetheless, it's around 125 or so. So this is on average. It can go up at times, it can go down at times. It depends on what's going on in your body. So how do we regulate it and try and maintain a fairly constant filtration rate? Well, there's three ways we do it. Something called renal autoregulation, neural regulation, typically from the sympathetic nervous system we learned in A&P 1, and then several different hormones that we've learned already we're going to revisit. So let's do renal autoregulation. First of all, there are two mechanisms that are considered to be autoregulatory. This word means that the kidney itself can control its own blood pressure no matter what the blood pressure is doing in the body. So for instance, if our systemic blood pressure goes up, that increased blood pressure ultimately will make its way to the kidney and thus may increase GFR. So the kidney can detect that and then try to bring GFR back down. On the other hand, and GFR is glomerular filtration rate. On the other hand, if the blood pressure in the system is too low, that low blood pressure is going to reach the kidney, which means it's going to make GFR go down. The kidney has the ability to detect, hey, the blood pressure is too low. GFR just went down. We need to increase pressure in the kidney to increase, specifically in a glomerulus, to increase GFR. All right. Are you guys kind of understanding that so far?
Yeah.
Okay. If you ever have a question, you can y'all know you know how to unmute yourselves, right? You can unmute yourself and then talk or type in the box. All right. So let's talk about this blood pressure regulation. The first mechanism that's autoregulatory is called the myogenic mechanism. This is the one that happens in seconds. Every second of every day, your afferent arteriole is increasing a little bit in diameter, but decreasing a little bit in diameter, increasing and decreasing in order to maintain that constant blood pressure inside the glomerulus. So it goes up, it dilates a little bit, it constricts a little bit, dilates a little bit, constricts a little bit, all day long. That's called the myogenic mechanism, and this is how it works. Let's say that the blood pressure in the body, the systemic blood pressure, that's what I'm calling it here, the blood pressure in the body goes up. That increased blood pressure ultimately will make its way to the afferent arteriole. And that high blood pressure stretches the wall of the afferent arteriole. Now, the afferent arteriole assumes that when it's being stretched, that means the pressure is too high. So the automatic response of the smooth muscle in the afferent arteriole, when it's stretched, is to contract. And when it contracts, it brings about vasoconstriction. And when you vasoconstrict the afferent arteriole, make it smaller, less blood comes in, you decrease the pressure and in the glomerulus, and the GFR goes back down to normal. Now, this is the quickest mechanism that happens to help adjust GFR by adjusting glomerular blood pressure. The smooth muscle will either contract if the pressure is too high, or it will relax if the pressure is too low. So that's a common theme. If we need to increase GFR, we want to vasodilate the afferent arteriole. That increases the volume of blood that gets in, you increase the pressure, and you filter faster. If, on the other hand, we need to decrease GFR, you want to vasoconstrict the afferent arteriole, which lets less blood in, which decreases pressure, and then you filter more slowly. And so that's one way that we're controlling how fast we're filtering and thus how fast the filtrate moves through the tube. All right. So that's fairly quick, fairly simple, easy way the kidney does it. It's automatic. We also have something called the tubular glomerular feedback mechanism. And the tubular glomerular feedback involves a feedback from, oh, that's not the picture that has it in it. This one does. It's a feedback from a special set of cells that's in the ascending portion of the nephron loop. They're called the macula densa. So I need to tell you about this little area right here, where the ascending portion of the nephron loop physically makes contact with the afferent arteriole during development of the baby in utero. The cells on that side of the tube become specialized, and they're called the macula densa. The macula densa are salt detectors. They have the job of detecting salt concentration in the filtrate as it moves past this part of the tube. On the other hand, the smooth muscle cells in the wall of the afferent arteriole, that are directly adjacent to the tube that is touching them, those smooth muscle cells become specialized. Now, at the very beginning of the semester, we talked about these cells. I called them juxtaglomerular cells, but I never told you where they were. Here is where they're located. The juxtaglomerular cells are the cells, also called JG cells, are the cells that produce renin. So if you remember, we talked about the renin, angiotensin, aldosterone system, which increases blood pressure in our body. Well, these are the cells that produce renin, all right? They're called the JG cells. So together, these cells, the macula densa and the JG cells, form what is called the juxtaglomerular apparatus. So what I want to tell you is how these macula densa cells are working right here. But before I go down there and do that, I want to go back up to the nephron picture so we can visualize where we're talking about. So the macula densa cells are in this part of the tube right there, where it would make contact with the afferent arteriole. Here's the afferent arteriole. It's going to carry blood into the glomerulus, and here's the ascending nephron loop. So where the ascending nephron loop is contacting the afferent arteriole, that's where the macula densa cells are inside the tube. So the reason why they're important is this. They detect the concentration of sodium chloride in the filtrate. So here's how easy this is. The faster we filter, the faster the filtrate will move through the tube. That's logical. If it's moving too fast through the tube, that means you have less time to reabsorb salt back into the blood. So if it's moving too quickly, by the time that filtrate reaches this part of the tube, there's too much salt in it. And the macula densa says, hey, wait a minute. There is too much sodium chloride in a filtrate. There's only one reason that would happen, and that's because we're filtering too quickly. So that's what the macula densa does. It's going to detect how much salt is in the tube as it approaches the macula densa. So what happens then? Well, the macula densa detects that high salt concentration, and it knows then that we're filtering too quickly. So the way that we reduce GFR, one way, is by decreasing glomerular blood pressure. So how do we decrease glomerular blood pressure? But nothing more than vasoconstricting the afferent arteriole. So the reason why it vasoconstricts is because the macula densa blocks the release of a vasodilator called nitric oxide. In the absence of nitric oxide, the vasodilator, when it's gone, the afferent arteriole will constrict, which increases blood pressure, which decreases blood pressure and decreases GFR back down to normal. So remember, I just mentioned the fact that normally the afferent arteriole is always larger in diameter than the efferent arteriole. The reason for that is there's a constant release of nitric oxide, which relaxes the smooth muscles in its wall and makes it bigger in diameter. So if you want to vasoconstrict this afferent arteriole, all you have to do is block the release of its vasodilator, which is nitric oxide. So in the absence of nitric oxide, the afferent arteriole vasoconstricts, it allows less blood volume in, it decreases the pressure in the glomerulus, and you start filtering more slowly, and sooner or later, the filtrate starts moving through the tube at its normal rate, and you start reabsorbing your salt, and by the time it reaches the macula densa again, the macula densa says, hey, the salt concentration is normal. So we can stop blocking the release of nitric oxide. And so the loop just turns off. So these are two autoregulatory mechanisms that will automatically control blood pressure in the glomerulus. Either bring it down to decrease GFR. On the other hand, it can go up. It can increase it because we can vasodilate that afferent arteriole. For the most part, I'm going to go through the hormones in a minute that regulate it as well, but the hormonal regulation is not considered autoregulation. Only these two mechanisms are autoregulatory. Now, there's a flow chart here that describes This mechanism that we just went through, you can view that later, talks about the salt concentration, the juxtaglomerular apparatus with the macula densa, decreases nitric oxide. You vasoconstrict the afferent arteriole, which decreases blood pressure in the glomerulus, and you start filtering more slowly. So that's the whole point of this autoregulatory mechanism. Now, as far as the sympathetic nervous system is concerned, the sympathetic system is what supplies sympathetic neurons to the blood vessels in the kidney. They don't get supplied by the parasympathetic system. So when you are physically active, or you're running on a treadmill, or if you remember from A&P1, the fight or flight response, the sympathetic nervous system fires when you're either scared, you're in an emergency situation, you're very excited, or if there's physical trauma to the body, like hemorrhaging. Oh, I'll put it in here. Like when you're losing blood, anything that's distressing the body or stressing the body, you have sympathetic output. Sympathetic output goes to the afferent arteriole and makes it contract. Now, we're not going to relearn the receptors on the smooth muscles there that we did in A&P1, but those were excitatory alpha adrenergic receptors. So when sympathetic nervous system fires, it basically constricts the afferent arteriole. And when you vasoconstrict the afferent arteriole, you decrease glomerular blood pressure, which decreases filtration rate. And when you decrease filtration rate, go back because I want to mention that as well. When you decrease filtration rate, you decrease the filtrate flow rate through the tube, and thus you're going to be having more time to reabsorb everything. And when you're working out, or if you're hemorrhaging or any other problem that you have the sympathetic nervous system firing, your kidney, the nephrons, will reabsorb your salt and water and put it back into the blood, almost all of it. So during a fight or flight response, you can stop producing urine altogether. But ultimately, with a mild fight or flight, you just reduce the amount of urine you produce because the nephron's reabsorbing everything. So you actually, during a sympathetic event, you're decreasing the urinary output volume, and it saves all of the water for the blood to maintain blood pressure. Remember, blood pressure is directly related to blood volume. So if we need to maintain or increase pressure, we don't want to lose water in urine. We want to conserve it and put it back in the blood. So during a sympathetic event, our afferent arteriole contracts, decreases pressure in the glomerulus, you filter more slowly, and the filtrate moves slowly through the tube, and you have more time to reclaim all your water and your salt and put it back in the blood. And that increases blood volume, but decreases urinary output volume. So I hope that makes sense, because urinary output volume and your blood volume are related. We increase our blood volume by decreasing urinary output volume, if you're not taking any more fluids in, of course, and vice versa. If you increase urinary output volume, you're decreasing your blood volume. And that's why doctors put people that have high blood pressure on diuretics. They want their kidneys to dump water out, bring their blood volume down to bring their blood pressure back down. So that's the idea behind that little bit of physiology. All right, so let's talk about these hormones. There are a couple of important hormones. We talked about them before. Angiotensin II, if you remember that name, and atrial natriuretic peptide. Now, in all of angiotensin II's effects, because there's 5 principal effects in the body, out of all of those effects, angiotensin II tries to increase your blood pressure, blood volume and blood pressure. So angiotensin II is always produced when our body is trying to increase blood pressure. And it's produced during the renin, angiotensin, aldosterone pathway. So one of angiotensin's roles is to decrease GFR. So let's go back to this picture up here. If I need to try and increase my blood pressure in my body, one thing, and this is only one thing that I know I don't want to happen is I don't want to lose my water in urine. So I need to try in some way prevent the loss of water in urine and put the majority of all the water that's being filtered out back into the blood. Angiotensin 2 actually helps that happen in a couple of ways. One of them is angiotensin 2 is a strong vasoconstrictor. So angiotensin 2 constricts really the apharyngeal as well as the epharyngeal. So by constricting these arterioles, you dramatically reduce the pressure in the glomerulus, which slows down the filtrate flow to a crawl. And it's going so slowly through the tube that all the tubule cells are reabsorbing almost all of the water. At times when we need to maintain blood pressure or increase our pressure. So angiotensin 2 is going to help increase our blood volume, which increases pressure by preventing the loss of water in urine. It doesn't control water reabsorption directly. It alters the pressure in the kidney, in the glomerulus, which slows down the filtrate flow rate through the tube, filtration rate, and thus flow rate through the tube. So angiotensin 2 is going to decrease GFR, which allows us to save our salt and water and basically increases our blood pressure. Now, atrial natriuretic peptide, ANP, is exactly different. ANP is released by special cells in the posterior wall of the right atrium in the heart. And ANP is released when the blood volume in our body is too high. So if our blood volume, meaning our water volume in our blood, is too high, that means your pressure is too high. So when you have too much blood volume, when that blood reaches the right atrium, it stretches it. And when the wall of the right atrium is overly stretched, it causes for the release of this peptide hormone, atrial natriuretic peptide. So atrial natriuretic peptide does a couple of things for us dealing with the kidney. It targets the kidney. One thing that it does is it's going to increase glomerular filtration rate. And it really does that, well, it increases glomerular filtration rate a little bit differently than what I've mentioned thus far. So let me explain that other way I mentioned earlier about how we filter, what controls filtration. So far we talked about blood pressure in the glomerulus. The blood pressure, if it goes up, you filter faster. If the blood pressure in the glomerulus goes down, you filter more slowly. And that's all regulated by the diameter of the apharyngeal. But there's a second way that we can alter the filtration rate without even adjusting the blood pressure. And that has to do with, which I don't show it too good in here, these little bitty cells up here that are in between the capillary loops. They're called mesangial cells. These mesangial cells have the job of lying against the capillary wall. And where they are lying against or touching the capillary wall, you can't filter through that spot. They basically block filtration where they touch the capillary. So if you could get these mesangial cells to move out of the way of the capillary wall, you have more space to filter through. So that's called surface area for filtration. So there's two ways we can alter filtration. You could change the blood pressure up or down, or you can change the surface area of the capillary, which is available for filtration to occur in. So I know I mentioned it in my video. If you haven't seen it, you should look at it. I came up with an analogy to describe this mechanism. So you have to know what a soaker hose is in your garden. A soaker hose is a hose that when you turn the water on, water seeps out the hose all the way down the length of the hose. So in my analogy, the soaker hose is the capillary loops, all right? And that's basically what we want. We're trying to filter out each part of the capillary. Now, imagine that you take that soaker hose and you wrap it up with duct tape in 1 foot sections, but then leave a foot open. You tape up a foot, you leave a foot open, you tape up a foot. All the way down the length of the hose, you have spots where you have duct tape and spots where the duct tape is missing. So when you turn the water on, The places where the water can only exit the hose is the open places where you don't have any duct tape. So in my analogy, the duct tape are the mesangial cells. So the mesangial cells have the ability to block certain sections of the capillary wall, so we can't filter right there. Now, imagine if you then removed all of the duct tape from your garden hose, your soaker hose. Now you turn the water on, and now more water is going to leave because you have water leaving down the entire length of the hose. That means you're filtering more. So what then removes the duct tape from your hose, or what removes the mesangial cells from the wall of the capillary? Well, lo and behold, it is atrial natriuretic peptide. Atrial natriuretic peptide is what causes the mesangial cells to move out of the way of the capillary surface, which increases the surface area and thus increases GFR. All right, so before we move on, does anybody have any questions about that particular mechanism? Because that does confuse some students about this surface area for filtration. Does anybody have any questions? No? I'm assuming everybody can still hear me, huh?
Yes,
Yeah.
sir.
Okay, good. So is everybody good to go forward?
Yeah.
Okay. All right, so I'll put in here a table that describes the different regulation mechanisms for GFR. Just go through their major stimuli and the mechanism of action and what happens to GFR. For instance, we just went over all of these, and you can see at the end over here what happens to GFR and what's causing it to happen. So make sure you review that. It's what I just talked about in those other slides. All right, now, the last thing I really want to cover in this chapter and get into chapter 27 a little bit before our time runs out is reabsorption routes. How do we reabsorb things in a tube, and how do we secrete things? So basically what we're about to talk about is how substances, fluids, and other substances are going to go from the middle of the tube back into the blood, called tubular reabsorption, and how substances will go from the blood back into the tube. Because it looks pretty easy. We could just say, hey, you know, fluid and solutes go from the filtrate straight through there into the blood, or vice versa. Well, it's not automatic. There are special transporters that have to operate in order for substances to either be reabsorbed or to be secreted. So ultimately, reabsorption mechanisms occur in two main ways. The substances that are being reabsorbed, here they show sodium. Sodium can be reabsorbed, and other substances, not just sodium. Substances that are reabsorbed can go between the cells that line the tube. So the majority of the cells, although we're not going through the histological changes, are cuboidal cells, and these cells line the tube. Over here would be the middle of the tube where the filtrate would flow down the tube. Here's on the outside of the tube in the interstitial space, and then the capillary where we're going to reabsorb and secrete from, back and forth. So substances can be reabsorbed between the tubule cells. If they go via that route, it's called paracellular reabsorption. Paracellular means on the side of the cell. That's why it's called that. Or the substances can go from one end of the cell through the cell and out the back end or lateral sides of the cell. So if you remember, this is called the apical membrane. These are called the basolateral membranes. And so when substances move from the apical membrane through the cell and then back out to get into the blood, it's called transcellular reabsorption. The prefix trans means through. So transcellular means through the cell. So what I want to explain to you all are some of the transporters that I'm going to have some questions on, that I want you to know what they're transporting and in which direction. But before I do that, I want you to look at this purple arrow that's showing sodium moving. You have to remember that any cell in the body that opens a sodium channel, when you open a sodium channel, it automatically wants to go into the cell, to the cytoplasm, down its concentration gradient. So when you open a channel, say right here, even though they don't show the channel yet. Sodium will always want to come into the cell because there's more sodium on the outside of cells than on the inside. So what we have is an unequal distribution of transporters along the membrane of this cell. And what I mean to say by that is this: at the apical edge of the cell and along a little part of the lateral part, you may have these sodium channels and other sodium transporters, which means sodium is always going to want to come in the front end of the cell. But at the basolateral membranes, you have those sodium potassium pumps. If you remember that name from a long time ago in general biology, these are active transporters that use ATP energy. And for every ATP that they use, even though they don't show it, you're going to pump 3 sodium ions out, but you're going to pump 2 potassium ions in. So the sodium potassium pumps are always running in the cell so that it keeps a one-way flow of sodium from the front to the back, from the front to the back, and back into the blood. Now, the reason why this sodium current is so important is because there are several different substances that will be reabsorbed along with sodium. So basically, sodium is controlling the reabsorption of other substances like glucose or sugar, some vitamins, amino acids, other substances, other electrolytes for that matter. So sodium's coming in. It's what I like to call the sodium current, and other things can piggyback a ride down the current so it can get into the blood. Even though it doesn't occur exactly like that, I always say it like that to make it seem a little easier. So the sodium balance from the outside to the inside and back to the outside is going to be important, because we're going to regulate the reabsorption of many things using sodium. So there are 2 main types of transporters that I'm about to talk about that involve the movement of sodium and other substances. If a transporter in the membrane, and these are all transmembrane proteins, if the transporter in the membrane allows 2 or more substances to move in the same direction through the cell, they're generically called a symporter. So the prefix SYM in Latin means the same. So symporters transport substances in the same direction, whereas antiporters transport substances in the opposite direction. So before we talk about those transporters, let me tell you about water, because I need to tell you how water is being reabsorbed as well. Now, I don't know if you ever heard me say it or other people say it, but at the end of the video or the packet or something, I put in there like, wherever sodium goes, water is sure to follow. Well, it's like that with a lot of solutes, but water always tries to go and dilute solute somewhere. So wherever sodium's going, it's making the solution in that water compartment a little bit more concentrated. So water is going to follow the sodium. So about 90% of all the water that's reabsorbed in the nephron is reabsorbed via what's called obligatory water reabsorption. That means that water is obliged or has to follow the other solutes that are being reabsorbed. So about 90%. The other 10% or so, maybe just a little more, is actually controlled by antidiuretic hormone. Antidiuretic hormone, if you remember, is the hormone that makes our kidneys save water and puts it back into the blood. So ADH tries to increase blood volume by decreasing water loss in the kidney. So in the absence of ADH, your kidneys are going to dump out 10% more water than it normally would, and thus your urinary output volume goes up and your blood volume would go down. On the other hand, if we have ADH, we reabsorb that extra 10%, and our urinary output volume is what, is normal output volume is, about a quart of urine a day, and we're maintaining that water into the blood. All right, so that's how we reabsorb water, and we're going to talk about that again in a minute. So there are different transporters in different parts of the tube. So the transporters I'm going to mention first are the ones, just a couple, are the ones that are in the proximal convoluted tubule. So, and I'll have questions on this. What are they? What are they transporting? In what direction are the substances moving? Things like that. So let's look at it. These transporters, at least 2 of them. So here is one of the transporters. This transporter is called the sodium glucose symporter. It's called the sodium glucose symporter because it allows sodium to come into the cell and for sugar, glucose, to piggyback a ride into the cell as well from the filtrate. So since sodium and glucose are moving in the same direction, I have to call that the sodium glucose symporter. So in this way, we're going to reabsorb sodium. We're going to pump it back out the basolateral membrane so that we reabsorb some sodium into the blood. And we have special glucose transporters that are isolated in the basolateral membrane. These are special glucose transporters. There's 4 different subtypes of them. But I will say this, the only place in the renal tubule, the only place, which let me go back up to this picture and make it a little easier. The only place in this entire renal tubule right here that contains the transporters which have the ability to reabsorb sugar is in the proximal convoluted tubule only. Once the filtrate goes beyond the proximal tube, if there's any sugar left in the filtrate, it's going to be lost in urine because there's no other place down the length of this tube that has the ability to reabsorb sugar. And I'm sure everybody knows that sugar in the urine is abnormal. And it typically occurs in people that have uncontrolled diabetes mellitus. And I'm going to explain to you why that is. So where's my picture? Here it is. All right, so here's our glucose transporters. We have the symporter, we got the GLUT transporter at the back, and we're reabsorbing sugar. Now, in people that have high blood sugar, and anywhere the maximum concentration is up towards about 160 milligrams per mil. But if you are approaching 200 milligrams per milliliter of blood, that is extremely high. And so what this means is that if you have too much sugar in the blood, you're going to overload the transporters. They won't have time to reabsorb all the sugar before the filtrate passes the proximal tube. So in that case, you're going to have what's called glucosuria. This is sugar in the urine. So if we go back to this picture, these transporters have a maximal rate at which they can reabsorb. So if you have too much sugar, some sugar is going to go in, but some sugar is going to bypass the transporter. So if you're starting with too much sugar in the blood already, that means you filter out too much sugar and you don't have enough time to reclaim it. And you're going to lose some in your urine, which is abnormal. You're not supposed to have sugar in the urine, all right? So people that have uncontrolled diabetes mellitus, meaning their blood sugar levels are not regulated appropriately because they don't have proper insulin function in the body, then they're going to filter out way too much sugar and they're going to lose sugar in the blood. I mean, in the urine, I'm sorry, because they have too much sugar in the blood, they're filtering too much out. Now, the other thing about people that have uncontrolled diabetes mellitus that you may already know is that often they urinate more than they're supposed to. They always have the urge to go to the bathroom and they're always thirsty. So why is that? Well, it's because there's too much sugar in the filtrate. And since there's too much sugar in the filtrate, just like I said, water is going to follow sodium. Water is going to follow that sugar. So people that are dumping sugar out in urine are also dumping more water out than normal because the water is following the sugar. All right, I hope that makes a little bit of sense. All right, so let's go and look at our other transporters. We're still in the proximal convoluted tubule. Here's just another picture of the transporters I just showed you with the reabsorbing sugar. Over here, though, we have the second type of transporter I want you to learn. It's called the sodium, and you might not be able to see it too well, but that's called the sodium hydrogen antiporter. This transporter allows sodium to come in. It will always want to come in passively through the channel or transporter here, but this specialized transporter allows a hydrogen ion to leave. As the sodium comes in, hydrogen leaves. Sodium comes in, a hydrogen leaves. Now, hydrogen is acid. This is This is what makes fluids acidic, is a hydrogen ion. So if you remember the carbonic acid cycle, CO2 and water combine together, forms carbonic acid, which breaks up into the acid component, which is hydrogen. That's going to be secreted out into the filtrate. But the bicarbonate will be reabsorbed back into the blood, and bicarbonate is one of the major buffers in the blood to buffer acid. So our kidneys are attuned to regulating blood pH because of a few things. This is just one of them. It has the ability to secrete acid out in what will become urine, and it has the ability to reabsorb bicarbonate, which can buffer acid in the blood directly. So you're getting rid of acid from the blood, and you're reabsorbing bicarbonate. All right. And we're going to get into that in a second a little bit more. Since I'm running short on time, I wanted to get through the rest of this because I want to teach you about acid-base balance as well. All right. So this next transporter that I want you to learn is found in the thick ascending limb of the loop of Henle, or in the ascending nephron loop. So that's the ascending part up here of the nephron loop. So in the ascending limb of the nephron loop, the more superior portion of it, it's outlined really thick right here, dark, because these cells in that part of the tube are impermeable for water movement. So only a little bit of water can get through whatever channels are open at that time, because the membrane is impermeable for water movement. So there are certain sections in the tube where they're more permeable for water movement. The proximal tube is very permeable. The descending tube is permeable. The ascending tube is not permeable for water. The distal tube allows a little bit of water depending on what hormone is present that I'm about to tell you about. And same thing for the collecting duct. So there are certain places where water is freely reabsorbed and places where we have to control its reabsorption. So the thick ascending limb of the loop of Henle is not the place where we control water reabsorption, but the important thing that happens in that part of the tube is the reabsorption of ions or salt. So remember, the macula densa is up here. So as the filtrate moves through this tube, if it's moving too fast, this symporter right here can't work correctly to reabsorb salt back into the blood, and the salt is bypassing the transporter and is too concentrated. That's when the macula densa believes that the filtration rate is too high. That explanation that we went through a while ago. So the transporter that's important in the ascending limb is called the sodium chloride potassium symporter. It allows a sodium with 2 chlorides to come in along with potassium. So potassium normally would want to leave the cell. Potassium is going against its gradient right here. But notice we have leakage channels at the surface of the cell. So potassium can leak, some of it can leak back out into the filtrate. And in that way, we have the ability to get rid of excess potassium. We don't want too much potassium being reabsorbed back into our blood, because too high of potassium, called hyperkalemia, can make you have a heart attack. So this is one way that we can help get rid of potassium out of the blood. I'm going to show you another way in a second. So at the basolateral membranes, we then reabsorb the sodium and the chloride back out into the blood. And that way you're taking salt from the filtrate and you're putting it back into the blood. So basically you're reabsorbing salt with a little bit of water that can get through here, but not much. All right, so let's talk about the distal convoluted tubule. The distal convoluted tubule still is involved in the reabsorption of sodium and chloride via symporters. But the added information that we need to look at is where we reabsorb calcium. Now, calcium can basically be controlled for reabsorption by parathyroid hormone. If you remember, that hormone increases calcium reabsorption in the kidney. Parathyroid hormone is released when our calcium levels in our blood are too low. And if your blood calcium levels are too low, the last thing you want to do is lose calcium in urine. So PTH targets the kidney, specifically the cells in the distal convoluted tubule, and then it causes for more calcium to be reabsorbed back into the blood, and basically prevents its loss in urine. Now, the distal convoluted tubule is slightly permeable to water, but it's not very permeable to water. So this part of the tube can be regulated for the reabsorption of water via hormones that we have to talk about. And in order to do that, I have to mention the types of cells that are in the last part of the distal convoluted tube and in the collecting duct. So let me show you where we're at on this nephron picture for this little bit of talk right here. So what we're talking about, the cells that exist in this part of the tube up here, the distal tube, and then what's called a collecting duct. So there's two different types of cells that line this tube at that point. So the cells that line the tube at that point are called the principal cells and the intercalated cells. The principal cells are the ones that are the most numerous. They're involved in reabsorbing sodium and secreting potassium out in urine. That's their main role. But they also are involved in a reabsorption of some water, and I'll show you that. The intercalated cells are involved in controlling how much potassium is reabsorbed, how much bicarbonate is reabsorbed, and how much hydrogen can be secreted in the urine. So I'll put in here two pictures that'll help explain this a little bit for you. So first of all, these are the principal cells that line the distal portion of the distal tube and the collecting duct. So some of the main transporters that are in here deal with sodium channels, potassium channels, and those sodium potassium pumps that I mentioned earlier. So these cells are sensitive, or I should say, have receptors for aldosterone. Aldosterone is produced by the zona glomerulosa of the adrenal cortex in response to two different types of stimulation. One, those glomerulosa cells release aldosterone if angiotensin II is present. And remember, angiotensin II tries to increase your blood volume and pressure. So another way that it does that is by causing aldosterone to be released. So aldosterone will be secreted. It will cause these cells to incorporate more channels and transporters in their membrane because aldosterone is a steroid hormone. It acts on the inside of the cell. So if you're increasing the number of these transporters, you're going to reabsorb more sodium and you're going to secrete back out more potassium. Now, the reason why that's important is because we don't want too much potassium getting back in our blood, but also it's logical because of the second stimuli that causes aldosterone to be released. The second stimulation that causes aldosterone to be released is a high level of potassium in the blood or interstitial fluid. So if you have too much potassium, that's called hyperkalemia. And during a hyperkalemic event, the zona glomerulosal cells will release aldosterone because it'll go to the principal cells and cause more potassium to be lost in urine. So we could bring the levels back down to normal. So these cells are responsible for controlling sodium and potassium secretion and then reabsorption for sodium. And remember, wherever sodium goes, water is sure to follow. So aldosterone basically makes these cells reabsorb sodium and indirectly then reabsorb water. So aldosterone, angiotensin II, and antidiuretic hormone, all three of those hormones try to make you increase blood volume and pressure by affecting the kidney in a different way. Antidiuretic hormone does so by making the incorporation of water channels. I think I'm going to show you that in a second. So special types of water channels that called aquaporin 2 channels that makes the cells more permeable for water reabsorption. So before we do ADH, let's talk about the other type of cell that is in the distal tube and in the collecting duct, and those are the intercalated cells. Now, the most important thing about the intercalated cells is that these are the cells that have the ability to increase the acid secretion or H secretion into urine. They do so more efficiently than other parts of the tube because intercalated cells have active transporters that use ATP energy to pump hydrogen ions up their concentration gradient about 1,000-fold. So that means that these cells have the ability to get rid of acid from the blood and decrease the urine pH more so than other parts of the tube. So the way that the kidneys do this by controlling blood pH is this. If our blood pH is too low, that means we're acidic. That means that the kidney will excrete more acid out in the form of hydrogen in urine, but it will reabsorb more buffer. So when your pH is low, you want more buffer to buffer the acid that'll bring the pH back up. And you also want to get rid of acid out in urine. So this is typically why also urine has a lower pH than our blood. Normally, it's like that. So people that then have some sort of an acidosis event in their blood, their kidneys are one of 2 organ systems in the body that can regulate blood pH at the organ level. And so the kidneys, again, want to regulate blood pH by either excreting more acid out and reabsorbing more buffer. That's if your pH is low. Or on the other hand, if our blood pH was too high and we were alkaline, we want the kidney to reabsorb less buffer. You don't want to reabsorb buffer if your pH is already too high. Too high. You don't want to reabsorb buffer. And also you don't want to excrete out your acid as much. You don't want to lose any more acid, and you don't want to reabsorb buffer if your blood pH is too high. That's the bottom line. And vice versa. And so I'm going to show you that in a minute. So in here, in intercalated cells, CO2 and water combine to form this carbonic acid. It splits up into hydrogen and bicarbonate. The hydrogen is the acid. It's actively pumped up its concentration gradient, and the bicarbonate is reabsorbed through what's called a chloride bicarbonate exchanger. It basically is an antiporter. So as we reabsorb one bicarbonate, which is an anion, a negatively charged ion, we have to reabsorb one chloride to balance off the difference in charge moving across the membrane. So this is called the chloride bicarbonate exchanger or antiporter. So they help regulate blood pH, right? Now, the other thing, there's also buffering systems in urine. So when we go to the bathroom, we're not urinating out free hydrogen or free acid. That would be bad. So the acids are actually buffered in the urine by either ammonia, which is NH3, that can combine to a hydrogen. And when NH3, which is ammonia, binds a hydrogen, it forms an ion called the ammonium ion, NH4, which is a positive ion, cation. So when that happens, that free acid, that free hydrogen now is bound and buffered and being carried out in urine bound to a nitrogen molecule, which we call ammonium ion. The other buffer molecule in urine is phosphates. So there's a buffering system called the monohydrogen phosphate. That's just one H on it. Phosphate is PO4. So this is monohydrogen phosphate. This is dihydrogen phosphate. Notice they have 2 H's on it. So a monohydrogen phosphate can bind to a free hydrogen, and it forms this dihydrogen phosphate, which then is carrying, both of these molecules are carrying acid out in urine. So that's what's explained on this chart. Now, since I can't draw out in front of you what deamination is, I can only explain it. Like, where does ammonia come from? It's a poison substance that we make in our body, and we have to get rid of it in what we call nitrogenous waste products. Well, if you look at a picture in your book somewhere or another book, or you can look it up on Google, what the structure of an amino acid is. An amino acid has 2 major groups on it. The front end of an amino acid has an NH2 group on it, which is called the amino group. On the back end of an amino acid, they have a C, a carbon with an oxygen and a hydrogen. That's called a carboxyl group. So when in our liver and other cells in our body modify amino acids, they chop off that amino group from the amino acid, and it binds to another hydrogen, which forms an NH3. So every time your cell cuts that amino acid in half, it's producing ammonia. which everybody knows is toxic because you clean with it at home. So we have to detoxify that from our blood. So our liver has the ability to grab those ammonia compounds and convert them to a less toxic form, nitrogenous waste, something called urea. So urea is really the nitrogenous waste that is coming from the breakdown of protein and amino acids. All right. All right. So let's talk about, just briefly, ADH. All right. So ADH, antidiuretic hormone, is going to increase water reabsorption in the kidney. It is a water conservation hormone. It's released from the posterior pituitary gland, if you forgot that. And it's released from the posterior pituitary gland because of special receptors in the hypothalamus called osmoreceptors that have the job of monitoring the concentration of solutes in the blood. So basically, if you're dehydrated, you don't have enough water in the blood, that means your solute concentration is high. So when you're dehydrated, the osmoreceptors say, hey, the osmolarity of the blood is too high. We need to save water and try and dilute the blood a little bit. So what does it do? It causes ADH to be released from the posterior pituitary gland. ADH targets the nephron, specifically the principal cells that I just showed you in the distal tube and the collecting duct. And so what ADH does to those cells is it causes them to incorporate water channels in their membrane. So ADH actually causes the cells to make a water channel that allows water to move from the filtrate back into the blood. The water channels are called aquaporin 2 channels. Now, there are aquaporin 1 channels, but they're found in the proximal tube. The aquaporin 2 channels are the ones that we can increase and decrease in number in the cell's membrane. So in the presence of ADH, basically all of the principal cells are more permeable to water. And so we reabsorb more water. In the absence of ADH, the cells are less permeable to water and more water will be lost in urine because you're not reabsorbing it. All right, so that's the loop for water regulation, reabsorption, and thus to control the water level in our plasma and other fluid compartments. This is a table included in here that goes over the major stimuli and effects of the hormones that we just covered. So it's all in one place. What we covered, you can just review that. Help you remember it just before you take a test. Now, before I go into the acid-base balance real quick, let's talk about what diuretics are. So antidiuretic hormone, ADH, tries to make you save water, puts it back in the blood, which decreases urinary output volume. However, there are different types of diuretics. In all cases, no matter how they're operating, there's different modes of operation or modes of physiological function. Diuretics, in some form or fashion, makes the kidney dump water out in urine. So you basically reabsorb less water back into the blood. So caffeine is a diuretic, but caffeine is a diuretic because it blocks the reabsorption of sodium. And if you're blocking the reabsorption of sodium, that means you're going to urinate more sodium out. And remember, wherever sodium goes, water is sure to follow. So caffeine basically makes your kidneys dump sodium and water out in urine. Alcohol is a diuretic, but it acts slightly differently. So when you drink alcoholic beverages, you might notice you have to go to the bathroom more often. That's because alcohol blocks the release of ADH from the posterior pituitary gland. And in the absence of ADH, your kidneys are going to dump out 10% more water than normal because of a blockage of facultative water reabsorption. And so that's what alcohol is doing. Now, all the different types of prescription medications in some way block the reabsorption of solutes in the renal tubule, either in the PCT, in the loop of Henle, or in the DCT. Some act at multiple spots. So the prescription meds are acting on the tube and blocking the cells from reabsorbing sodium, similar to what caffeine is doing. Now, some of them have, you know, effects on potassium channels as well. Like there are potassium sparing diuretics. That's going to make the person not lose all of their potassium through a diuretic, which I really don't have time to get into that. But nonetheless, all the diuretics alter the reabsorption of an ion or a solute, and it makes the water dump out in urine. So diuretics increase urinary output volume, but it decreases blood volume. And for that reason, the first mode of medical therapy for hypertension, you get put on a diuretic so that you lose water in urine and it brings your blood volume down and hopefully your pressure down. The majority of the time, people are on diuretics as well as other meds to help control their blood pressure. All right, so that's it for that chapter. I know I've kept you for a while. If you have something to do, you can leave the room if you want, but because you can always come back. This thing's recording, I believe. I hope it's still recording. And you can just watch it later on if you want, because I know I've been talking for a while, but I definitely wanted to get to acid-base balance. All right. So can everybody still hear me, by the way? Somebody can unmute and talk to me.
Yes, sir.
Okay. Thank you, Lena. All right. So chapter 27 is fairly straightforward. I will say this. Chapter 26 has a note packet. So you need to read through the note packet as well as the PowerPoint. Chapter 27 questions are going to come straight from the PowerPoint because I didn't generate a note packet for this chapter. So I'm going to use the information that's in this chapter. So for the most part, the chapter, can y'all see the new chapter on the screen, by the way? Chapter 27.
What's it called?
Okay, do you see, it's chapter 27, fluid, electrolyte, and acid-base balance. Or do
No, sir.
You don't see it? All right, I got to close this one then first. Hold on. All right, uh-oh. All right, can you see it now?
No, sir.
Oh, you don't see it? Oh, my screen sharing is paused. That's why. Hold on.
It just says diuretics.
Okay, that's why. Hold on, I'm trying to figure this out. Why is my screen share, new share? All right, can you see it now?
Yes.
Yes, sir.
It's a chapter 27?
Yes, sir.
Okay, good. So evidently when I switch out my app, it pauses screen sharing. So everybody can see the new one now, right?
Yes, sir.
Okay, good. All right, so as I was saying, I want you to just make sure you go through this. It talks about water balance, where the fluid compartments are, and water shifts a little bit. Most of this stuff is self-explanatory. Some of it coincides with what I just talked about with the kidney. I put in here, again, what some of these hormones are doing and the mechanism and all of that. So just review it. I also want you to know what water intoxication is. Water intoxication is where someone has too much water in their blood relative to the concentration of solutes. So yes, you can have too much water. And here's a little flow chart that describes how this can happen. So ultimately, you know, you can have excess blood loss via severe sweating, vomiting, diarrhea, things like that. Outside exercising drastically, and but the way that you replenish your water is you only drink plain water. This is not good. If you drink just plain water and you're moderately to severely dehydrated, what it does is it decreases the concentration of sodium in the plasma. Because now you put too much plain water in your plasma relative to the salt load in your blood or sodium load, and that then induces what's called hyponatremia. Hyponatremia is where you don't have enough, you have too much water and not enough salt in your blood. So what does that do anyway? Well, it decreases the osmolarity of your blood drastically. An increased osmolarity of your blood means you don't have enough water. A decreased osmolarity of your blood means you have too much water. So what happens when you have too much water in your blood? Well, let me go back to this picture real quick. Water always tries to go from one compartment to another one by diluting solutes. So basically, water will move from where there's more water to where there's less water. So if you have too much water in your blood, water is going to move to the extracellular space, the extracellular fluid. And from the extracellular fluid, it's going to go into your cell. And if a whole bunch of water is doing that, your cells begin to swell up. And that's exactly what we don't want to happen. Normally in our body, we want our concentration of the fluids to be about the same. We want them to be isotonic so that we have the same amount of influx and outflux, efflux of water. So the membranes don't get stretched and the cells don't pop open and die. Well, if you consider the cells in your brain, your neurons, the neurons in the brain start to swell up. And when that happens, it causes major confusion. It can prompt seizures. Person can fall down with major seizures. It can put someone into a coma and you can die from it. Now that is a very severe case of water intoxication. And it typically would happen in somebody that, say, runs a marathon, and at the end of the marathon, they just drink a gallon of plain water. That can begin the process of water intoxication. Now, this process is a little harder to ensue in an adult, a healthy adult, but it happens more easily and more quickly in babies and young children and elderly people. It happens more so in babies because their body to surface area, their volume to surface area ratio is very high. That means they have a lot of surface area relative to body mass. As we grow, we typically begin to have more body mass than surface area. So babies are more prone to having this. And that's why if you have a small baby, a small child that has a major fever and they're sick, you can't just give them ice water. You have to go give them Pedialyte because you're supposed to replace your water volume in your body with electrolytes and water, not just plain water. So that's what this little talk in that book is about. All right, so I need to get down to pH because I know we're carrying way over. Also through here, I talk about the electrolytes, where they're concentrated and all of that. Just review it. You guys know these ions already. So just review that for the test. Now, the acid-base balance. This little talk is going to help you for the lecture part as well as the lab part, okay? Blood pH range, normal range, is 735 to 745, something you already know. We talked about that briefly in the blood chapter. So whenever something happens to make our blood pH fall below 735, that means we are acidic and you are in what is called acidosis. On the other hand, if something is going on in our body that makes our pH go above 7.45, our blood pH is too alkaline, and in which case you are in what is called alkalosis. So in order to prevent large swings in pH changes of our blood, we have various mechanisms to control pH. We have buffering systems to help buffer the acid in our blood. Our lungs and our kidneys regulate pH at an organ level. So we have buffering systems in our body. Our lungs help regulate pH by controlling how much carbon dioxide we exhale out. And our kidneys control pH in our blood by controlling really two things. I should have put the other one there. It controls how much acid we excrete out in urine, and it also controls how much buffer is reabsorbed. So let's go through the buffer systems briefly. You don't have to go and memorize these structures, all right? You just have to know what the buffer systems are. But I left this in here so you can see what's going on. First of all, a buffer, by definition, is any substance that can do two things. The buffer can either act as an acid, where it can release hydrogen into solution, and thus make the solution more acidic, or the buffer can act like a base and reabsorb acid out of solution and brings the pH back up. So a buffering molecule can actually either release hydrogen into solution, it makes the solution more acidic, or it could soak the hydrogen up out of solution, which can make the pH more basic. So what they're showing you here, this is the basic structure of an amino acid. Oh, I can tell you what happens with deamination. Here's the basic structure of an amino acid, which amino acids make up our proteins. When we are processing proteins in our liver and other cells, this group right here gets cut off. But the hydrogen on this back group over here, called the carboxyl group, binds to it, and that's what forms ammonia. So that's what I was talking about at the end of the chapter 26. But as far as a buffer is concerned, if the pH of the solution is too high, some amino acids can release this hydrogen into solution. And by doing that, it brings the pH of the solution back down. Well, we're really talking about our plasma, but it relates over to the other bodily fluids as well. So if our blood pH is too high, there's some amino acids on proteins that can release hydrogen in solution and bring the pH back down, and vice versa. If the pH was too low, some amino acids can take the hydrogen and combine it to the amino group, which forms that ammonia compound that I was just talking about. So it acts like a base. So this is what's called the protein buffering system, which includes proteins like albumin in our plasma, one of the plasma proteins. There's a whole bunch of protein in our plasma that have the ability to absorb or release hydrogen. And thus it's part of what we call the protein buffering system. Now, hemoglobin is in our red blood cells. It's also part of that protein buffering system, but I think this chapter kind of gives it its own class of buffer. But since it's a protein, I always lump it into the protein system. So hemoglobin has the ability to absorb acid. And here's what happens. If we forgot what happens in a red blood cell, I'll go over it off this picture. Now, all of this chemistry that you're looking at here is occurring on the inside of a red blood cell. So water and CO2 combine together to form carbonic acid in the presence of carbonic anhydrase. The enzyme that catalyzes this step is not shown, but it's called carbonic anhydrase. So it forms carbonic acid. Now, carbonic acid then splits up into a conjugate acid and base. So it releases hydrogen in solution, and it releases bicarbonate, which ultimately becomes an important buffer. So what happens to the hydrogen that's being dumped off of carbonic acid on the inside of the red blood cell? Well, it actually combines with the protein portion of hemoglobin. So that protein portion of hemoglobin absorbs or binds to the hydrogen ion, and it makes the hemoglobin acidic. Now, with that said, if you can remember back in the respiratory chapter, one of the parameters that decreases hemoglobin's affinity for oxygen is acid. So if hemoglobin becomes acidic because it's binding to hydrogen, that causes for more oxygen to be released from hemoglobin to go to the body cells to use it for aerobic respiration. So I hope this is kind of coming full circle now. So when our cells need more oxygen, it's because they're producing a lot more CO2 anyway via aerobic respiration. And that CO2 gets converted into a carbonic acid, which gets broken down into hydrogen and bicarbonate. The hydrogen gets absorbed on hemoglobin, which makes it acidic, which reduces hemoglobin's affinity for oxygen, but increases oxygen dissociation. So that's how all of that becomes full circle. And by the way, hemoglobin then becomes a buffer because it prevents the water in the red blood cell and thus around it to become acidic because you're holding on to the acid. That's what buffers do. So in this case, it's buffering that hydrogen. So it's part of the buffer system. Now, the carbonic acid buffering system occurs in different cells of the body as well as in red blood cells, but we use hemoglobin to bind to the hydrogen in red blood cells. Now, in the body, our lungs actually utilize this same exact chemistry and in our blood. So at what we call external respiration, if you remember that, at external respiration, hydrogen is going to combine with bicarbonate, which then forms carbonic acid. But since hydrogen is bound into this molecule, it actually brings your pH back up because it's not free in solution. It's being buffered by bicarbonate, is binding to it. Now, carbonic acid at external respiration doesn't just stay as an acid. It actually, oh, it's not the picture I wanted to see. Hold on, see if it, no, that's not it. I don't have it. So at external respiration, acid is going to combine with bicarbonate and then convert into carbonic acid. Now, this is the buffer, bicarbonate. It's held in place by this acid. And by removing hydrogen out of solution, you're bringing the pH back up. However, if we have to bring the pH back down, all this acid does is releases hydrogen back into solution. But before it does that, some of that carbonic acid is converted back into CO2 and water, which is not shown. So this reaction right here, where water combines with CO2 to form carbonic acid and then breaks down to hydrogen and bicarbonate, this reaction is reversible. So bicarbonate can combine with hydrogen, which reforms carbonic acid. And carbonic acid, when the blood is in our lung level, is broken back down to CO2 and water, and this is what we exhale out. So by you breathing deeper and faster, you're exhaling more CO2 out. Look what happens to the acid in the blood. The faster you exhale CO2 out, the faster this reaction runs in the reverse direction. So by running in the reverse direction, bicarbonate always combines with hydrogen to form carbonic acid, which splits up into CO2 and water, which you exhale out. And so we're going to talk about the lungs in a second. I'm going to clear that up if it doesn't make too much sense to you. The last of the buffering systems is the phosphate buffering system. That deals with dihydrogen phosphate and monohydrogen phosphate. You don't have to memorize this chemistry. I'm not making you go, we have to know this particular chemical reaction. You're just relearning it because we did that before. You don't have to go and memorize the phosphate buffering reaction. Just know that dihydrogen phosphate and monohydrogen phosphate are the ions used in the phosphate buffering system. And it can either absorb hydrogen or release hydrogen into solution. All right. Oh, here's what I was looking for right here. This is the reversible reaction for the way the lungs control blood pH. So let's go over this one more time because I know it was kind of hard going back and forth. So in the respiratory system and in our body, the direction in which this reaction occurs is based on the concentrations or how much of the molecules we have on either side of this reaction. So if we have more CO2, the reaction will run in this direction. If we have more hydrogen and bicarbonate, the reaction will run in this direction. So for that reason, at internal respiration, when we're trying to give oxygen to the cells from the blood and remove CO2 from the cells back to the blood, the cells are making a lot of CO2 inside our body that gets dumped into the blood. That CO2 goes into the red blood cell, combines with water to form carbonic acid, which splits up into hydrogen and bicarbonate. Then that bicarbonate gets back out into the blood, which I'm not going to re-show you. But when the blood re-enters the capillaries in the lungs, this reaction is reversed. So at external respiration, this reaction runs from right to left. Bicarbonate combines with hydrogen, which means it's removing hydrogen from solution and thus bringing the pH up. So bicarbonate combines with hydrogen, it forms carbonic acid, which splits up into water and CO2 by the action of carbonic anhydrase, which again is not shown. That's the enzyme that catalyzes it. And then you exhale this out. So let's say someone is acidic. For one of the reasons I'm about to talk about, because the kidneys aren't working or whatever the case may be, and their blood pH becomes acidic. If their lungs are working correctly, your patient will begin to respire more quickly and deeper. So when our blood pH starts to drop, we breathe deeper and faster in order for this reaction to go to the left more quickly. So by breathing deeper and faster, we're getting rid of CO2, getting rid of CO2, getting rid of CO2 as we exhale, but this reaction can keep running over there in that direction. So as you're running in that direction, look what's happening. You're removing hydrogen, you're removing hydrogen, you're removing hydrogen, and then exhaling the CO2 out. Excuse me. So the lungs help regulate blood pH at the organ level by controlling how fast we breathe in and how slowly we breathe in and out, and by how much CO2 we're exhaling out. So here is a little negative feedback loop for the respiratory center called the dorsal respiratory group in the medulla oblongata. And when we become acidic, we have a decrease in our blood pH, which means the hydrogen ion concentration is going up. Those receptors in the medulla oblongata that have the ability to detect pH are called central chemoreceptors. The central chemoreceptors basically monitor pH of cerebrospinal fluid. We also have chemoreceptors out in the periphery of the body. They're found at the carotid sinuses in your neck, just above your collarbone, and in the arch of the aorta. Those are called the peripheral chemoreceptors. So the peripheral chemoreceptors monitor the chemistry of the blood. The central chemoreceptors monitor the chemistry of cerebrospinal fluid. So if the blood pH is going down, so does the pH of the cerebrospinal fluid. So both of these receptors are going to fire to the dorsal respiratory group in the medulla oblongata. And these nerve impulses make the dorsal group neurons fire more quickly out to the diaphragm and the external intercostal muscles, which make you breathe faster and deeper, which means you exhale more CO2 out. And so you're going to keep doing that, breathing faster and deeper until you get rid of all your CO2. And then this reaction will begin to slow down in this direction so that we start to retain more of our hydrogen in our blood when our blood pH is back up to normal. So when we're acidic, we breathe fast more quickly and deeper, and the reaction runs to the left more quickly. If our blood pH was too high, it would be the exact opposite. You would want to retain as much CO2 as possible so the reaction runs in this direction. So let's talk about some of those events. All right. Oh, wait, I got to tell you about the kidney first. So the kidneys are the other organ in our body that regulates pH at the organ level. And it does so by doing two things. The kidneys' intercalated cells that we just covered have the ability to excrete a lot of acid or just a little bit of acid. It has the ability to reabsorb more buffer or less buffer. So I'll give you a simple example. If the blood pH is too low, you're in acidosis. We want the intercalated cells to reabsorb as much buffer as possible to buffer the acid in the blood and bring the pH up. But on the other hand, we also want the CO2 that can come out of the blood or in these cells to get rid of acid through the carbonic acid equation. This is a major way that we get rid of acid through a volatile acid, which carbon dioxide produces a volatile acid because it either goes back and forth to a gas. We can also get rid of hydrogen from a non-volatile acid, but nonetheless, the acid can be excreted out in urine more and concentrated more in the urine. So by excreting more acid out, you're removing acid from the blood. That'll bring the pH up. By reabsorbing more buffer, you bring the pH up. So that's what we want to happen if the pH is too low, right? So let's go and look at the acid base. Oh, this is just all the mechanisms that we just went through. Let's go through the 4 primary acid base imbalances and then we'll be done. I know y'all are probably tired of hearing me talk, but I wanted to get all of this done today and I didn't know I had this much to talk about. But nonetheless, there are 4 principal primary acid-base imbalances. First of all, any imbalance that brings you below 735 is called acidosis. Anything that makes you go above 745 is called alkalosis. Now, there are different reasons why the pH can become abnormally low or abnormally high. If the pH change is due to the fact that the respiratory system is not doing its job correctly, the acid-base balance would be called a respiratory one. For instance, if a person is not breathing, let's say you have a patient and they stop breathing, they might have hit their head, or maybe they have a pulmonary obstruction or something, something stuck in their trachea, they're not breathing. Or even if you hold your breath. If you hold your breath, then the respiratory system is not getting rid of CO2. So let me go back to this, might make it a little simpler. If you hold your breath, this reaction runs in this direction from left to right. In which case, as you're holding your breath or your patient's not breathing, CO2 is building up in the blood. And if CO2 builds up into the blood, you release a lot of acid. So that is what's going to bring your pH down. So if the person's blood pH is going down because they're not breathing, it would be called respiratory acidosis, right? Now, if their pH goes up and it's due to the fact that they're hyperventilating, that means you're getting rid of too much CO2. I'm sure everybody has experienced this before. If you ever hyperventilated before, you get kind of lightheaded. That's one of the signs and symptoms of an alkaline pH in your blood. So you actually induce respiratory alkalosis if you hyperventilate. So if we're breathing too quickly, then this reaction runs to the left. So if you're breathing really fast, you're running to the left, you're basically pulling hydrogen out of the blood, out of plasma, you're putting it back in the form of carbon dioxide and water, and you're exhaling the acid out. And so you're getting rid of too much hydrogen, your pH then goes up. If you are holding your breath, the reaction runs to the right, and you're producing more acid, so your pH goes down. So we have two principal respiratory issues. They're either called respiratory acidosis or respiratory alkalosis. The other acid-base imbalances are metabolic. So that's what we call metabolic acidosis or metabolic alkalosis. And these are due to the fact that the kidneys are not doing what they're supposed to be doing correctly, or some other condition is occurring in the body, altering pH, but it's not due to the lungs. We call that metabolic acidosis. And if the pH goes, what's that? Did somebody say something? Can y'all still hear me?
Yeah.
Oh, good. I heard something. I hope the meeting's not going to kick us off. I'm almost done. All right. So metabolic alkalosis is when the pH goes up, and metabolic acidosis is when the pH goes down, and it's due to the fact of how much bicarbonate we have. So if you don't have enough bicarbonate in your blood, you don't have enough buffer, your pH goes down. You become acidic. If you have too much buffer in the blood, you buffer too much acid and the pH goes up, and that's called metabolic alkalosis. So here's a quick table that I'll just tell you real quick because I just heard something. I think our meeting is maybe getting stopped or something. I don't know. All right. So the 4 primary acid-base imbalances, there's 2 main things you have to know about each one of them. Well, you have to know what some of the common causes are, but the 2 main things is how to recognize what the acid-base imbalance is, and that's what I wanted to get to. So the acid-base imbalance is either called respiratory or metabolic acidosis and alkalosis. It's based on the concentrations of really 2 things. How much carbon dioxide is in the blood means something. And the level of carbon dioxide in the blood is measured in millimeters of mercury of pressure, and it's represented by a pCO2. The other factor that's important about pH in the blood is bicarbonate. So we have to know what the normal ranges of CO2 and bicarbonate are in order to understand the pH imbalance. So for instance, the normal range of blood PCO2 is 35 millimeters of mercury of pressure to 45. So 35 to 45 is the normal range. So if you did what is called an ABG on somebody, which is an arterial blood gas, and you tested this PCO2, and the PCO2 was higher than 45 millimeters of mercury of pressure, then that person would be in respiratory acidosis. If you have too much CO2 in your blood, that means you're not exhaling it out. And it also means that the reaction is running to the right and you're dumping acid out in the blood. So for one of these main reasons over here, which I'm not going to go back down through them, you're not breathing correctly for whatever reason, your CO2 builds up in the blood, your reaction runs to the right, you're dumping acid in the blood, and you become acidic. So when the PCO2 is higher than 45, the condition would be called respiratory acidosis. On the other hand, if the PCO2 was lower than 35, then the condition is called respiratory alkalosis. That means somebody's breathing too fast, like hyperventilation, or if they're high up on a mountain and they're breathing fast because there's not enough oxygen, or pulmonary disease, or whatever the case may be. Then you're exhaling out too much CO2, so the PCO2 falls below 35 millimeters of mercury of pressure, and your pH goes up, which is called respiratory alkalosis. So to identify the condition, we have to know the PCO2. Now, to recognize if your patient is in a compensation, you have to know two things. One, you have to know what the pH is, and you have to know the concentration of CO2, at least in this condition. So in respiratory acidosis, in that imbalance or alkalosis, the organ that's going to try and compensate to bring the pH back to normal are the kidneys. So if a person is in a respiratory imbalance, the kidneys are going to try and do something to bring the pH back to normal. So let's start with this one. This is one of the easier cases. A person in respiratory acidosis has too much CO2. Their pH falls below 735. If the kidneys are working correctly over some amount of time, depending on how bad the condition is, the kidneys can bring the pH back within its normal range. And if the pH comes back within its normal range, even though the lungs are still not dumping out enough CO2, or the CO2 is still high, but the pH is normal. If the pH range is normal, but your CO2 levels are still high, that means a person has respiratory acidosis, but they're in renal compensation. That's what we say. Because the pH is back to normal, but their CO2 level is still a little high. It ultimately will come back down if you can correct whatever the problem is with the respiration. And so ultimately the PCO2 may come back down to normal if you're put the person on a ventilator, right? Sort of like, with this COVID thing going around, everybody's getting put on a ventilator, their lungs are malfunctioning. They can't get rid of enough CO2. They can't get enough oxygen. So people were dying. So that ventilator breathes for the person in and out, exhaling it and putting oxygen in the blood. So in some case, if someone hits their head on the back of their head where the medullary center is at, they might stop breathing. That person might be put on a ventilator until and if and ever when they can start breathing on their own again. So respiratory acidosis, too much CO2, pH falls, kidneys come in and reabsorb more bicarbonate and dump more acid out in urine, brings the pH back within normal ranges, you're in renal compensation. Respiratory alkalosis is the opposite. Your PCO2 levels fall below 35 millimeters of mercury of pressure, and when the CO2 levels fall too low, pH goes above 745. So that would be called respiratory alkalosis. In this condition, we want the kidneys to stop absorbing buffer and to stop excreting out acid in urine. Because if our pH is already too high, you don't want to buffer it. It's going to go up even more. And for that matter, you don't want to lose any more of your acid out in urine because it'll make the pH go up even more. So if you're in respiratory alkalosis, you know you're in renal compensation because the kidneys made the pH come back to normal and your CO2 levels are still a little low. So that's how you know you're moving into full compensation. When the pH is normal, but the CO2 is still low. All right. Now, if the pH is normal and the CO2 is normal, you don't have a acid-base imbalance and you're fully compensated. So we won't say respiratory alkalosis with renal compensation. You're just compensated because everything's back to normal. Now, as far as metabolic acidosis and alkalosis goes, it deals with bicarbonate. So there are different conditions whereby your bicarbonate levels can change and bounce out of their normal range. Bicarbonate is measured in what is called milliequivalents per liter in our blood. The range is from 22 to 26. So anything that makes your bicarbonate levels drop below 22, it makes your pH go down in the blood because basically you don't have enough buffer. So if you don't have enough buffer, then the pH is going to drop because you're not buffering it. So a loss of bicarbonate ions via severe diarrhea or the accumulation of non-volatile acids in the body. And what I mean to say by that, it can't be a CO2 issue. If it's a CO2 issue, then it's a respiratory condition. But if you know the term ketoacidosis, people that are diabetic, uncontrolled diabetes, they get ketoacidosis. It's the buildup of ketones in the blood. And it happens because our body burns fat in the absence, or lipids in the absence of sugar. So when ketones build up in the blood, you then go into ketosis. Ketosis is a decreased blood pH due to the buildup of ketones. And you might have heard of that term because of all those new diets. Ooh, I'm in ketosis. Well, I'm not going to get into all that, but it makes you burn fat. But it's not always good to just do it constantly because it drops your pH. So the buildup of non-volatile acids, acids that cannot be turned into a gas, or the loss of your bicarbonates directly can decrease our blood pH by decreasing our bicarbonate. So if your bicarbonate levels are low and your pH drops below 7.35, that's called metabolic acidosis. You know you're in respiratory compensation if your patient is breathing very quickly. So the lungs will all of a sudden try and compensate by hyperventilating. So somebody's in metabolic acidosis, the lungs are going to try and get rid of acid by getting rid of the volatile acid, converting it back into carbon dioxide and exhaling it out. So you know you're in metabolic acidosis if HCO3 is too low and pH is low. And you know if you're in respiratory compensation because the pH will go back into a normal range, but the bicarbonate will still be low. You're in respiratory compensation. Now, for metabolic alkalosis, this happens because of vomiting, gastric suctioning, pulling the gastric contents out, diuretics, or people that take too much of like milk of magnesia, or they eat too many Tums. Basically, you're getting rid of the acid out of your body. Some diuretics make the kidneys dump acid out. So that's why that's in there. But nonetheless, let's look at what happens if you void the stomach of all of its hydrochloric acid. When the stomach is losing its hydrochloric acid, either vomiting or suctioning or whatever the case may be, or you're buffering it with alkaline drugs like Tums and milk of magnesia or whatever, what the stomach lining does is it tries to produce more hydrochloric acid because you just removed it. Well, the byproduct of producing hydrochloric acid is the production of bicarbonate. So in the stomach lining, the acid that's being made goes to the lumen of the stomach, inside the stomach. But the bicarbonate side product goes into the blood. So by taking acid out of the stomach, you make the stomach make more acid, which indirectly makes more bicarbonate, the bicarbonate goes in the blood and you have too much of it now. So anything that makes your bicarbonate level go above 26 milliequivalents per liter, it will bring your pH up. Because you have too much buffer in the blood, you're buffering all the acid and your pH goes up. So that would be called metabolic alkalosis. Now, the lungs will try and compensate for the pH change. It's not going to do anything for bicarbonate, but it'll try and bring your pH back to normal. So if you're in metabolic alkalosis, your patient is going to start to hypoventilate. You breathe more slowly. That means you get rid of less CO2, and acid can start to build up in the blood. And it'll bring your pH back down to normal. So you know you're in metabolic alkalosis, bicarbonate's higher than 26, pH is high, and you're in respiratory compensation because the pH is normal, but your bicarbonate is still a little too high. All right, so I hope that helps a little bit. You can review the videos and the PowerPoints and all of that. I know I kept you about 2 hours now or so. So does anybody have any questions before I stop the meeting?
Mr. Russell, I don't have a question on this topic, but I have a question on something else.
Okay, what is it?
I was still wondering if you will be curving our unit test grade still.
I believe I went in and curved the lab grade so far in lab. I haven't had a chance to go look at our lecture exams yet because I've been real busy trying to help some of these other faculty members go online. So I'm just now getting to catch up with all of my own stuff.
Mm-hmm.
So yeah, I'm going to look at it. And if I see that it's bad, I'm going to curve it. So oh, look, I can see some people on there. That's cool. This thing is pretty cool. So yeah, I haven't had a chance to look at it yet, Lena, but I'll look at it.
Okay. Thank you, Mr. Russell.
Okay, you're welcome. All right. So before I stop the meeting, does anybody else have any questions?
I have a question too. I tried to log into the thing when you do the test, and it won't let me.
In lecture or lab?
In the lab, when you go to take the test that you got to be respondents. It froze.
It froze up?
Yeah, and it won't like go past a certain point. I see my face and then it won't go forward.
Okay. I'll go in there and reset. You should be able to do it as many times as you want. I keep trying,
and I get to a certain point and I freeze.
Okay, I think make sure your audio is working. There's a couple. I don't know how to fix it, but I have heard that if respondents cannot detect your audio, or the camera, it has to be able to. It has to be able to detect your camera and your audio. In order for you.
Today is working.
I know. I see that. What kind of computer do you have? It's not a Chromebook, is it?
Yeah.
It's a Chromebook?
Yeah.
That's why it's not going to work on there. Respondus does not work with
Oh, Jesus. Oh, okay, it's a Dell.
Oh, it's a Dell?
Yeah.
It's not a Chromebook then, right?
No, it's
a full-fledged laptop that you have a hard drive on there, you can generate documents and save it and all that?
Yeah.
Like a regular computer, right?
Uh-huh.
Okay, well, it should work on there. Tamika, I'll go in and look at your respondus thing. And what about the one for lecture? It works for the one for lecture?
I just tried to do the first one. That's the one that's not working for the lecture one.
Oh, it's not working for the lecture one?
Yeah.
Oh, I thought you said the lab. All right.
Yeah.
I'll go in and check it. I don't think I have to reset anything, but just try and do it again. The other thing you can do is use the help feature in Canvas. Have you tried that yet?
No.
To get the technical support people on it and see if they can figure it out.
All right.
And then just email me back because I'm going to be posting some announcements about the people that can't get Respondus to work.
Okay.
But if you've used Respondus on other tests, it should work.
Well, this will be my first time using it.
Yeah. So something's going on with it. You can just try it again. If I have to unlock it, then just I might not be able to get to it until later because this class went way over. I'm supposed to start another class at 1 o'clock.
Okay.
So, but the reason why I kept y'all on is because I definitely wanted to get through some of this material. But if you go to try it again and it says that you can't do it, just email me.
I will.
All right,
Tamika,
Tamika.
I had the exact same problem earlier this semester. What I had to do was just delete the respondus and re-download it, and then it
Oh
worked
yeah,
fine.
I forgot.
Okay, I'll try doing that.
Okay.
Thank you.
Thank
And
you
the
for that,
last other
Veronica.
question was that, you know, when we did those modules 1 through 6, do we get any credit for that or no? Because it's not on the.
In lab?
Yeah.
Yeah, you're going to get credit for all that stuff. What I'm going to do is compile all the new material. That's what you're talking about?
The old material.
Oh yeah, you're going to get credit. I haven't ported the grades over. I have to do that by hand.
Okay.
So basically what I have to do is go into the Engage site, and you're still supposed to be doing all those Engage quizzes.
Oh,
Are you still doing them?
I stopped after 6, I think.
Okay, well, they're all still open. They all are
Okay.
open until the 27th, I think.
Oh,
So,
God.
Yeah, you need.
Extend them. Extend them to next week. I'll do them.
Yeah, it's open. It's open through until next week.
Okay.
Because I think I opened all of them.
Okay.
Because those Engage quizzes are still going to go into our grade book, but I have to go grab all those by hand and manually put them in. And I usually wait until the end to do that.
Okay.
All right. If y'all have any other questions, please just email me so I can start getting ready for this other lecture. And I'll get back to you, and I'll post some announcements dealing with the final as well with instructions. Thank you all for coming into the room. I appreciate not talking to myself. Some other classes, I've only had one person, so I appreciate y'all being here. All right.
Thank you, sir.
All right. I'll talk to y'all later.
Y'all have a good week. Be
Good luck
safe.
studying.